Hey guys, welcome to our very last digital lecture. Um, wow. I just want to say, first and foremost, I miss you guys so much. What I wouldn't give to just be doing this in the classroom with you guys. I do. I can't even believe that we've been home for 2 months. But I do miss you guys terribly. Ugh, awful. Um, with all that being said, let's talk about plans for the last time. So part one was general anatomy. Part two, xylem, phloem. And now part three is more like the bigger picture mechanism of how stuff moves in the xylem versus the phloem. So first we have the phloem. Remember, phloem is food. So the big things that they're going to talk about is sucrose or amino acids. But most of the talking through and explanations and stuff is going to be just using sucrose as the easy example. So first of all, it's called translocation because you're translocating the sucrose from source to sink. And then this is a really, really, really big term, mass flow. It flows in a mass. Super random thing, but they love that word. Mass flow, mass flow, mass flow. So it all starts when we've got a source from the source, which is producing these red dots or sucrose. So the source is going to be a leaf or a storage organ or something that has a ton of sugar in it. It's going to diffuse sucrose into the companion cells, and then the companion cells can move it into the sieve elements. That can be passive or active depending on how much is where and what's needed. But there are proton pumps and co-transporter proteins, just active transport mechanisms, ready to do that. But it just kind of depends what the concentration is of what and where and when. So this is a big wow, that's why sieve elements have to be alive. That's why companion cells have to be alive, because sometimes these sugars, amino acids, whatever, assimilates, need to be pumped actively against the gradient, which means you need ATP, which means you need mitochondria, which means you need to be not dead. So trans mass flow, sucrose actively loaded, okay. And then the next big idea is that it's going from source to sink. So a source of sucrose is anywhere that is producing sugars, so super photosynthetic tissues where a lot of photosynthesis is happening, leaves mostly, or where sugar is stored. So if you have something like a potato, which is a tuber, that's storing sugar in the root of the plant. Potatoes have a ton of starch because starch is a sugar used for storage. So, but that's generally it. Leaves, or if there are storage organs in the roots. So from source, it's going to be pumped into the sieve elements from the source, and then it's going to move towards the sinks where there is less sugar, therefore those tissues need sugar. So sinks are roots and shoots. When roots are growing underground, they can't photosynthesize, but they still need to grow to support the plant. So that's a big sink of sucrose, of energy, is growing those roots. If you have a plant that looks exactly the same from the ground up, you don't necessarily know how much growth is happening down below. And then shoots, if you're growing a new leaf, if you're growing a flower, if you're growing a fruit, if you're growing something new, it may not be developed enough to start photosynthesizing, or it may be working on growing itself instead of photosynthesizing. So that can also act as a sink for sucrose. And that's where it's going to be removed from the sieve element wherever it needs to be. So source to sink. One thing I do want to point out is that in this example, the roots could be either. So if you have something like a potato or a storage organ in the root, that can be a source of sugar. It can also be a sink. So it depends on the context. There would have to be context there. I couldn't just say, are the roots a source or a sink? Because it could be either. Which also reminds us that there is two-way flow. So we saw last time that in the xylem, water only moves up. Sucrose, assimilates, sap can move up or down, just depending on what's needed where and when. Okay, and then so it is sap. It is think like maple syrup. It is to some extent a liquid. There's water involved. So there's the sugars, the everything else kind of dissolved in water. So there is also what's called a hydrostatic gradient, where there's water concentrations pulling it from high to low as well. So water is involved as well, but not to a super huge extent. But you do want to note that there is a hydrostatic gradient there. Okay, so loading sucrose in the phloem. Mesophils in the leaf produce a lot of sucrose from photosynthesis. I know sucrose is a disaccharide, but that just ends up being the main that main sugar that's moved and used in plants. So the mesophyll cells are especially photosynthetic in the leaf. So they're going to make it all, and then it's going to move across the mesophyll, eventually getting to the phloem down here. And that can be through the apoplastic or the symplastic route. We're not really concerned about how it's getting there at this point. So then sucrose is loaded into the companion cell against the concentration gradient most of the time. So there are co-transporter proteins, there are proton pumps, and then it's moving either. So this slide I should change a little bit. It really, really, really depends on the context. This is the typical situation. So typically you have to actively load it into the companion cell and then it passively flows into the sieve element. But in like ride or die situations, it could kind of flip that way and be like passively into one and actively into the other. But this is the typical, like most of the time it looks like this. So loaded into the companion cell with co-transporter proteins against the gradient, and then it flows down the gradient into the sieve tube. Unloading at the sinks. So at a sink, it's going to be a tissue that needs sugar. So it's going to be a tissue that's low in sugar. So it's going to act or passively just diffuse out of the sieve tube. Once it's there, the sucrose can be broken down by enzymes to use the monosaccharides involved, and that's going to decrease the concentration again. So it's going to keep that concentration gradient running in tissues that need sugar. So source to sink will still apply. Okay, so then, like we said, the xylem is one-way flow, roots to leaves, it evaporates from the leaves, and then more comes from the roots, and then it just keeps going up. So as water evaporates from the leaves, it creates a lower water potential in the leaves because it's evaporating, so less water in the leaves. It's going to keep it moving up. So this is also called mass flow. Mass flow applies to both. And then there's this cohesion tension theory, which is just the idea that water tension and cohesion and adhesion too is what's pulling the water up. So water is going to be moving up with the help of cohesion. So remember, we're going against gravity at this point. We're going up like a huge tree potentially. So hydrogen bonds, water pulling other water. Water also adheses really well to lignin. So the lignified cell walls and adhesion is helping as well. Well, GIF. So we have cohesion, we have capillary action, adhesion, and then there is some pressure in the roots too. So just properties of water, hydrogen bonds, water is getting pulled up. It works. We talked about this. So the water typically is going to follow this route into the root hairs, into the cortex, into the xylem, go up the stem, and then it's going to end up in the mesophyll of the leaves, where it's then going to probably evaporate out of the stomata. Remember that stomata are pores at the surface of a leaf that can open and close to regulate water intake and loss and gas exchange. So the problem is going to become that the stomata need to be open at certain times or for X amount of time to allow oxygen and carbon dioxide to diffuse in and out for photosynthesis. And with those gases, water vapor is going to escape as well. So it becomes kind of like a struggle back and forth, which is transpiration. So transpiration is the loss of water vapor from the leaves via evaporation. Transpiration is just evaporation through the leaves. So 90% of transpiration occurs through the stomata. So by a plant being able to open and close its stomata, it can control a good portion of the transpiration that's happening. There is that trade-off of gas exchange, but the control is there. And then transpiration pull. So transpiration pull is the idea that transpiration is a driving force behind the water movement in plants. Because transpiration is occurring, because water is evaporating from the leaves, there is a lower water concentration in the leaves. When there's a high water concentration in the roots and a low water concentration in the leaves, the roots are going to bring in water and it's going to end up traveling towards the leaves. So it's the idea that that removal of water from the leaves is going to keep driving the movement of water up, and that whole cycle is driven by transpiration. Another thing that they say that they really emphasize is that transpiration is an inevitable consequence of gas exchange, which is just the idea that because you have to have oxygen and carbon dioxide coming in and out for photosynthesis, water vapor is also a gas, it's also going to escape. So because stomata open and close and have to open and close at certain times for photosynthesis to occur, the transpiration is going to occur too. So if it's a really hot, sunny day, there's a ton of photosynthesis happening, and that leaf needs to do gas exchange. It has to decide, is it worth losing that water to get those gases? So it's, it's a back and forth, an inevitable consequence. Here, let me move myself. This is just kind of a big picture. So there's a higher water potential in the roots, lower in the leaves, and water molecules in the xylem flow down that concentration gradient. So low to high, thinking back to diffusion, it's still, working here. So transpiration cohesion hypothesis. Oh, this kind of brings it all together. So we're intaking water from the soil. There's cohesion in the xylem, adhesion in the xylem, and then evaporation or transpiration, to be more specific, is the driving force that's driving everything else pulling upwards. Okay, so this is more like investigative stuff. So measuring transpiration rate. Like we said, most water vapor is lost through the stomata, and stomata can open or close. So you can investigate the transpiration rate at under different conditions with this thing called a potometer. We're going to do an online lab with this, which is so exciting. But the idea behind a potometer is that if you have a piece of plant, you can seal it into this tube, and the tube has water in it, and a calibrated pipette so you can see the measurements. So as the plant takes up water, as transpiration occurs, so as transpiration will occur, more water is going to get driven up, and then you can see the water level is going to move down as the plant takes it up. So the more water that's absorbed through the plant, you can assume the more water is lost via transpiration. So factors affecting transpiration rate. These are all fairly common sense, but you need to know every single one of them. So if you didn't print out the notes, maybe jot some of this stuff down. So different things can affect transpiration rate. Think about just like a plant outside in the sun for all this. So light, when you have a really bright sunny day, the stomata are typically going to need to be open for photosynthesis to occur. If you have a lot of sunlight, photosynthesis is going to be very good. It's going to be happening regularly. So the stomata are probably going to have to open for gas exchange to help with the photosynthesis. So more light is going to increase transpiration. At the same time, most plants will close their stomata at night because there's no sunlight, there's no photosynthesis. They're allowed to kind of save the water. So transpiration will go down and darker if it's darker at night. Temperature, so think about just water evaporating. Higher temperatures, more evaporation. Higher temperatures also help to dissipate humidity, even though it might not feel like it in South Florida sometimes. Humidity, so humidity is going to decrease the water potential gradient between the leaf and the air outside. If you've got 80% humidity outside of the leaf and 70% humidity with water vapor inside the leaf, then it's not going to move as quickly or as much as if it were really, really dry outside with more water vapor on the inside of the leaf. So humidity decreases transpiration. Wind, so especially when you have this kind of like bubble of still air around a leaf when transpiration is happening, it kind of leads to this like little bubble of humidity. It can increase the humidity around the plant. So wind kind of dissipates all that. It's hard for it to be humid when it's windy because it keeps everything moving. So wind is going to tend to increase transpiration. And then the last one is soil water. If there's more water in the soil, the plant's probably taking up more water. It's probably losing more water. So yeah, you need to, if you were given a scenario, you would have to describe what's going to happen to the transpiration rate and how or why that's going to happen. So these are really important. And then the very last thing is xerophytes. So xerophytes are things like succulents and cacti. They are plants that possess physical modifications to their structure to reduce water loss. Big, super duper important distinction here is that xerophytes are not the same as C4 or CAM plants. So C4 and CAM plants may be xerophytes, but xerophytes are not necessarily C4 or CAM plants. So C4 and CAM plants, yeah, I'm sure you guys learned about those in AP, but that has to do with photosynthesis and how that plant goes about photosynthesizing. So unrelated. Xerophytes just have certain physical modifications to help prevent transpiration. C4 and CAM plants are doing photosynthesis differently. Not to say that xerophytes probably use those, but they're not overlapping. It's like, I don't even know. I can't think of an analogy off the top of my head. Not the same though. Xerophytes are just physical adaptations. Just because something has powdery leaves doesn't mean it's a CAM plant, essentially. So lists of adaptations, you should be able to recognize these in a picture. So if I gave you a picture of a succulent, it was like, what xerophytic adaptations does this have? You need to so xerophytic adaptations, just a list of adaptations. Smaller leaves, smaller surface area, less diffusion. So that could be things like pine needles. Plants can have a thicker cuticle layer that's going to help reduce loss through the cuticle. Waxy or powdery deposits on the leaves. That kind of helps with like buildup of localized humidity / stop, kind of blocks the stomata to an extent. They may just have less stomata. The stomata can be concentrated on the bottom of the leaf. If the top of the leaf is facing the sun, it's going to be warmer, it's going to be lighter. So bottom of the leaf's a little bit more protected. Sometimes stomata can be like sunk into like these pits or like these deep pores. And that's also going to kind of protect it from like the light and the temperature outside. Rolled leaves. So sometimes they kind of roll in towards each other, also protecting the stomata. We can also have epidermal hairs or scales to trap moisture around the stomata. So this is kind of a blurry picture, but like those fuzzy succulents, those hairs help to trap moisture, and more moisture, more humidity outside the leaf is going to decrease transpiration. So here's a picture of a leaf. Just some examples. You guys are going to do practice problems with this, but this one, for example, has a rolled leaf. It's got these hairs. It's got, we can't really see the cuticle here, stomata and pits. Here's a real picture that looks kind of like a bug. Bet it's a monococcus. It's got faces. So that one also, that cuticle is pretty thick. You can see it pretty well. Those hairs, those deep pits for stomata, but they end up all looking pretty similar. So you guys will practice with some of this, but just be aware of like, if you see it, be able to identify it as far as xerophytic adaptation. Oh, that's it. Okay. We're all done now. Fin.